Hi everyone, I'm Tengfei Xue, a PhD student from Harvard Medical School and the University of Sydney. Glad to present our work in ISBI 2022. I'm going to introduce a deep learning-based framework for tritography pixelation of superficial white matter. Diffusion MRI tritography maps brain white matter connections. The whole brain tritography contains hundreds of thousands of streamlines. A streamline is a sequence of points along the curve produced by tritography. It estimates the trajectory of white matter tract in the brain. However, tritography is not directly useful. White matter pixelation is needed for quantification, visualization, and neuroscientific studies in health and disease. As shown here, it identifies tritography streamlines into anatomically meaningful tracts. Traditional tritography pixelation methods using either ROI-based selection or streamline clustering. Recently, some deep learning-based methods have been used for tritography pixelation successfully. However, most methods focus on deep white matter. Few methods can pixelate superficial white matter. Superficial white matter, SWM, includes short-range U fibers connecting adjacent and nearby gyri. The figure shows some U fibers in human brain. The volume of SWM is twice as large as DWM in our brain. The SWM pixelation is challenging due to the small size of U fiber connections and high variability across brains. No deep learning methods have yet focused on SWM pixelation. We propose the first deep learning-based SWM pixelation method, superficial white matter analysis, SupWMA. It is trained on a high-quality large-scale labeled streamline dataset. The neural network is point cloud-based. Since tritography streamlines are sequences of 3D points, point clouds can potentially enable efficient representations for streamlines. Our training procedure includes two phases: supervised constructive learning and downstream learning. Supervised constructive learning allows the network to obtain more discriminative features between plausible streamlines and outliers. SupWMA enables SWM pixelation of whole brain tritography across populations and diffusion MRI acquisitions. The high-quality large-scale streamline dataset was derived from an anatomically curated atlas. It includes SWM clusters and DWM clusters. The atlas was curated from high-resolution diffusion MRI data of 100 young healthy adults in Human Kinetome Project (HCP), and the atlas was labeled by a neuroanatomist. Our dataset has 1 million streamlines and consists of 198 SWM clusters and 1 non-SWM cluster. Outlier streamlines are also included in the dataset. We use this labeled dataset for training and validation. We use point cloud-based network for streamline pixelation. The point cloud-based network can efficiently encode streamline points into latent space. The network can also be invariant to point orders. That is, streamlines with equivalent forward and reverse point orders can have the same global feature. For example, streamlines from cortex and subcortical structures or vice versa can be represented equally. In addition, because location in the brain is important for streamline classification, we remove transformation nets from PointNet to preserve the pose and orientation information of streamline. The network also becomes more lightweight, it saves the training time, and streamline pixelation is faster. The network is shown here. It has two parts: encoder and classifier. Encoder takes 15 streamline point coordinates. Each point is encoded individually by the 3 MLP and aggregated by the max pooling to get the streamline global feature. The classifier takes the global feature to output the class label for each streamline. Identifying plausible and outlier SWM streamlines is still challenging, even with the efficient point cloud-based network, because many SWM plausible and outlier streamlines can have high geometrical similarity. The figure here shows an example. Although the outlier has a sharp bend in its curve, they are almost in the same location and size. Point coordinates along these two streamlines are very similar. They are highly likely to have similar global features in the latent space of the network encoder output. Therefore, we use supervised constructive learning (SCL) to help the encoder extract more discriminative global features for streamlines. SCL extends self-supervised constructive learning by proposing a supervised constructive loss. The training procedure has two phases. In phase 1, the encoder and the projector are together trained with supervised constructive loss. The projector has two fully connected layers. It can benefit downstream tasks by retaining more streamline information into the global feature. In each training batch, streamlines with the theme label are positive pairs, and streamlines with the different labels are negative pairs. The constructive loss will pull global features of positive pairs closer and push apart negative pairs. In phase 2, we freeze parameters of the encoder and discard the projector. The classifier is trained with cross-entropy loss for streamline classification. The figure here shows the process of SWM parcellation with trained SupWMA. The unlabeled tritography data of a subject is shown on the left in blue. It is registered into the training data space. In registration, we use the B0 image of the subject and the T2me image of atlas populations. Then the trained model takes streamline features of the transformed tritography to predict the class label for each streamline. The parcellation results with 198 SWM clusters are shown on the right. For evaluating our method, we tested on the expert label dataset and 3 independently acquired datasets. This slide shows the results of five-fold cross-validation on the expert label streamline dataset. The bar graph on the left shows accuracy, which is defined as the percentage of streamlines that are classified into correct labels. The middle bar graph shows F1 score, which can reflect both precision and recall values. Macro F1 score calculates the average F1 scores across classes. It can better show the performance of our unbalanced dataset. The bar graph on the right illustrates the floating point operations, FLOPs, which measure the number of required operations for model inference. It's used to evaluate the efficiency of each method. We compared the proposed method with other two state-of-the-art deep learning methods. Both methods are designed for DWM parcellation using CNNs and streamline spatial coordinate features. We train them on the label datasets based on their codes. As in this figure, SupWMA in the red bar achieves highest accuracy, macro F1 score, and lowest FLOPs. This demonstrates high performance and efficiency of SupWMA. We also evaluate the proposed method on 3 independently acquired datasets across populations and different MRI acquisitions. HCP is a dataset with high-quality different MRI data from young healthy adults. Adolescent brain cognitive development (ABCD) includes different MRI data from more than 10,000 American children. Parkinson's progression markers initiative (PPMI) dataset contains healthy older adults and older adults with Parkinson's disease. These datasets were acquired from different image protocols. We use 100 subjects from HCP, 25 from ABCD, and 25 from PPMI. We add a clustering-based method WMA for comparison. WMA uses spectral clustering for parcellation. The unification rate is the percentage of 198 SWM clusters that are detected in each subject, and we measure the mean and standard deviation of this rate as in this figure. A cluster is considered to be detected if it contains at least 20 streamlines. SupWMA achieves highest class unification rate across datasets. It demonstrates that it has good generalization to testing datasets. This is a visualization of an example SWM cluster across datasets and methods. The cluster we choose here is located in the frontal lobe. SWMA identifies more streamlines that are plausible than other state-of-the-art methods on these example clusters. In conclusion, we propose SupWMA, a novel deep learning framework for SWM parcellation with successful applications across populations and different MR acquisitions. SupWMA employs a modified point cloud-based network and uses supervised constructive learning to improve white matter parcellation tasks. Compared to state-of-the-art methods, SupWMA is the top performer on an expert label dataset and 3 dependently acquired testing datasets. Thank you for listening.